Welcome everyone to today's webinar, Shifting the Paradigm in Cancer Screening, a multi-cancer early detection testing. On behalf of Becker's Healthcare, welcome, and thank you so much for tuning in. We are thrilled to be joined today by Dr. Tom Baer. Dr. Baer is the Chief Medical Officer and Vice President of Multi-Cancer Early Detection at Exact Sciences and an Adjunct Professor of Medicine at OHSU Knight Cancer Institute. Dr. Baer, thank you so much for joining us today. I'll now turn the floor over to you.
It is such a pleasure to be with you today to discuss multi-cancer early detection, also known as MCED or MCED. I'd like to start with a brief review of my disclosures. In addition to being an employee of Exact Sciences, prior to joining Exact Sciences, which is coming up on a year now, I was an active investigator at OHSU, working with a variety of companies in the multi-cancer early detection area, and those are listed here on this slide. I'd like to start with the why of what we're working on. As we all know, cancer is one of the main public health threats that we all face. It is the second leading cause of death behind cardiovascular disease, and early detection is the best way to make a difference. But today we have consensus guideline recommended screening or early detection tests for only 4 common cancers. And that leaves 2/3 of incident cancers and 2/3 of cancer deaths due to cancers for which we do not have available early detection technologies. Multi-cancer early detection is the most straightforward and realistic way of addressing this major public health gap. We see MCED as a strategy to markedly broaden the range of screen-detected cancers, enable the early detection of cancer at early stages when intervention can make a difference, and markedly expanding access to early detection to all Americans. Now, the effort at MCED test development at Exact Sciences is a long-standing one. Began in the laboratories at Johns Hopkins University looking at 2 biomarker class approaches to cancer early detection, and that led to DETECT-A, a large feasibility study that I'll be talking about in the next several slides. That study was published in 2020, and since then the teams have been hard at work adding technology from Mayo Clinic laboratories and Exact Sciences laboratories to develop what is now a 4 biomarker class multi-cancer early detection test that's nearing clinical trial testing. Now, I'd like to start with a review of DETECT-A. This was a landmark study, the first large-scale prospective interventional evaluation of a multi-cancer early detection test in the United States. It relied on a 2 biomarker class design, an earlier version of the test we're working on today, and it enrolled over 10,000 women between the ages of 65 and 75, the only criterion being that they could not have a prior history of cancer. This study was conducted within the Geisinger Health System. As a result of MCED testing and over the ensuing 12 months of follow-up, nearly 100 cancers were diagnosed in these 10,000 women. On the left-hand side, you can see the distribution of cancers by how they were diagnosed. 24 of them were diagnosed as a consequence of standard of care screening test, which of course continued as part of routine healthcare. An additional 26 were detected as a result of MCED testing. So a single blood test detected as many, if not slightly more cancers than all standard of care screening tests combined. And an additional 46 presented clinically, and we'll go over those in a moment. On the right-hand side, you can see the distribution of stages of those cancers detected as a result of an MCED blood test. And so you can see that all 4 stages were represented and a number of cancers were diagnosed at early stages. Here's a summary of the cancers that were detected as a result of an MCED test. And you can see that they included some cancers for which standard of care screening tests are available, for example, breast, colorectal, and lung, although I will note that recommended screening for lung cancer is limited to individuals at elevated risk as a result of a smoking history. But there are also a number of cancers like ovarian, thyroid, uterine, appendiceal, and kidney, for which we do not currently have screening tests. And these were also picked up through an MCED test. Notably, the DETECT-A study also served as a proving ground for our diagnostic resolution strategy. We have developed an approach that we believe is straightforward and definitive, and relies on imaging to resolve a positive MCED test result. Patients undergo an IV contrast CT and PET scan when they have a positive MCED, and then the additional diagnostic procedures as necessary are dictated by the results of those imaging studies, as well as the clinical judgment of the healthcare provider evaluating that patient. At ASCO in 2023, we were able to present long-term follow-up results from DETECT-A. This is a unique data set in the field because this study was the first such study. It's the only study that now has multi-year follow-up, where we're able to take a look at the outcomes in patients whose cancer was diagnosed as a result of an MCED, and also look at what happened with those individuals who received a false positive result. We now have 4.3 years of follow-up from the initial test for both of those groups. First, looking at the patients whose cancer was diagnosed directly as a result of an MCED test, we took a look at this through a variety of lenses. So on the left-hand side of a slide, you can see that there is a very strong relationship between favorable outcomes and the ability to offer surgical treatment. 54% of patients with an MCED-detected cancer were treated surgically, and 86% of those patients are alive and cancer-free at last follow-up. Now, of course, being able to offer surgical therapy was strongly related to early-stage diagnosis, and the results were even more impressive in individuals who had a cancer detected at stage 1 or stage 2. There, 100% of patients who were treated remained cancer-free at last follow-up. For patients with a stage 3 cancer, the results were not as good as stage 1 and 2, but even in those groups, what we're seeing is 60% of individuals are still alive, and then more than a third of those patients are alive and cancer-free when they were detected at stage 3. Turning our attention to false positive patients, these are individuals who had a positive MCED test, but an imaging and clinical evaluation that did not reveal a cancer. And I get a lot of questions about this scenario. There's a lot of concern out there that having a false positive MCED result might identify an individual at high risk who needs to be followed closely and who may be quite worried about what to expect in the ensuing years. So we're really happy to be able to take a look at actual outcomes in individuals like this over a 4-year period. And what we found is that 98% of individuals with a false positive result remained cancer-free over the ensuing 4.3 years of follow-up. 2 out of the 98 individuals had a cancer diagnosed. They were diagnosed between 2.5 and 3 years after their initial testing. And one of those patients was a very early stage breast cancer diagnosed on mammography. The other was a stage 3C ovarian cancer that was very aggressive in nature and claimed the life of that woman within a year of diagnosis. We don't know for sure that these were new cancers and they were not missed by the MCED, but the clinical characteristics of them lead us to think that they may very well have been. And importantly, with 2 cancers diagnosed over 4 years in nearly 100 women, the annual risk of cancer was well below 1%, which is a level of risk that one would expect in women aged 65 to 75. So at least in this data set, in DETECT-A, it looks like our diagnostic strategy is able to give us a robust resolution. And for those individuals who have a false positive result, they can be reassured that their residual risk of cancer is no different than that expected for their age group, and is not elevated and should not require intensive or unusual follow-up strategies. So in summary, the results from DETECT-A first demonstrated that an early version of the Exact Sciences MCED was able to double the number of cancers detected as a result of screening, including a number of cancers for which we don't currently have recommended screening tests. We're able to see now with longer-term follow-up that when cancers are detected with an MCED test at early stages, the outcomes are exactly as we would hope. For stage 1 and 2 patients who are treated, 100% of them are alive and cancer-free. These are preliminary and relatively small numbers, but really quite encouraging as we head into larger prospective studies of our next generation MCED test. And importantly, our imaging-based diagnostic strategy appears to yield informative results that can provide appropriate reassurance to those individuals who have an initial positive MCED test but are later classified as false positive. The residual risk of cancer in those patients appears to be no higher and maybe even lower than what would be expected for that age group at baseline. So these are the results of DETECT-A, now turning the attention to our latest efforts to further improve the performance of the MCED test under development, and a bit about our next steps. So what I've shared with you are the results of a 2-biomarker class test that was evaluated between 2017 and 2019. Since that time, the team has been hard at work to further improve the performance of our test. And to do so, 2 additional biomarker classes were added, methylation and aneuploidy. So the test that we're currently working on involves 4 different biomarker classes being evaluated. We examined the impact of moving to 3 biomarkers and then to 4 biomarkers in a large case control study involving more than 4,000 samples that were divided into a training and validation set and into an independent test set where the performance of our assay under development was evaluated. I'll summarize here the performance in the test set. First, we looked at a 3 biomarker class test, and that revealed an overall sensitivity for a broad range of cancers of 55.2%. And that's across 15 cancer types at all 4 stages. When we added the 4th biomarker to our test, the overall sensitivity improved to 62.4%. The important finding here, however, is not the overall sensitivity, but where that gain in sensitivity was seen. And the greatest gain is seen in the earliest stages of cancer. We were able to achieve an overall sensitivity above 40% for stage 1 and stage 2 cancers combined. That's an unprecedented level of sensitivity for MCED tests under development and provides us a good deal of confidence as we move forward towards large-scale clinical trials. So what we took away from this work is that a 4 biomarker class test that involves aneuploidy, methylation, mutation, and protein biomarkers results in a high sensitivity for multiple cancers as a consequence of utilizing complementary signals from a variety of biologic signals that cancers secrete into the blood. We measured the performance of this test against cancer types that represent 75% of all cancer deaths. And we found that the test was capable of detecting signal from all of those cancers across all stages. Now, I do want to say that the test is not being designed for that 15 cancer type set. That just happened to be the cancer types against which its performance was measured. The cancer test that we're designing relies on shared cancer signals from a broad range of cancers, and we expect it to be capable of detecting a broad range of cancers. In the next case control study, we'll be looking beyond the initial 15 cancer types to evaluate its performance. And the overall sensitivity of 62% is really a pace-setting level of sensitivity for the field as we head into the next round of studies. So we see the future of cancer screening as a complementary approach between standard of care testing that we have available today that's proven to save lives, and a robust multi-cancer early detection effort that complements that standard of care approach to broadly expand the range of cancers that we can screen for and the accessibility of screening to all US population segments. Currently, we're finalizing the design of our test, and we seek to initiate a large-scale randomized pivotal clinical trial of our test that will be designed to seek FDA approval for our test. And it will be complemented by ongoing additional case control and real-world evidence efforts. We're going to do this in collaboration with a broad range of stakeholders. We're in close communication with the National Cancer Institute, with a number of scientific and research groups, investigators, medical centers that will collaborate with us in our clinical trials efforts, as well as a broad range of cancer advocacy groups and policymakers, all of whom are working together to make multi-cancer early detection a reality so that we can markedly reduce the morbidity and mortality from cancer. And as I conclude this talk, I wanted to bring to your attention one of our collaborators. That's the Multi-Cancer Early Detection Consortium. The website is shown here, mced.info. This is a unique organization that brings together companies working to develop MCED tests, academicians involved in this space, advocacy organizations, research organizations, as well as payers, hospitals, and health systems to really sketch out a way forward to make MCED a reality. So in summary today, I think I showed you that in our large pilot study, a prototype early version of our MCED test more than doubled the number of cancers detected with screening. We've shown you now with long-term outcomes that when cancers are detected early with an MCED test, outcomes are extraordinarily promising. And when cancers are not detected, but a positive signal is seen, our diagnostic evaluation strategy can conclusively rule out the presence of cancer and leave individuals with a high level of confidence that their residual risk of cancer is no different than it would have been for the similar age group without the test result or the workup, and that no special follow-up should be necessary. And finally, I've shared with you some data about our continued development and improvement of our MCED test, and updated you on the fact that we're moving forward with a 4 biomarker class design into the final stages of our test validation, and shortly afterwards, a large-scale pivotal randomized trial. With that, I'd like to thank you very much for your attention, and I welcome any questions from the audience.
Thank you so much, Dr. Baer, for all of that information. We do have a few questions that we would love to touch on today from the audience. So our first question is, how are you addressing population diversity with the Exact Sciences multi-biomarker MCED test?
Yeah, thank you for that question. So, I think for tests like the MCED test, diversity from the very beginning is critically important. So if you think about why enrollment of diverse populations into clinical trials is important. For drugs, for example, it's important because we need to understand if a particular drug or cancer treatment regimen works equally well in all groups in our population. But if a study doesn't meet its diversity goals, one can go back and do additional studies to close those gaps. That's not desirable, but it can be done. For tests like the MCED test, in the case control studies that we're doing, we're actually developing the test. We're using the information from individuals with cancer and without cancer to lock down the thresholds for our biomarker assessment and the algorithm that determines the cancer and no cancer signal. And so it's critically important that we include everyone from the very beginning, because you don't get to redesign the test easily. So at Exact Sciences, we've been deeply committed to enrolling a diverse group of patients, not just into our upcoming pivotal trial, but also into the case control study that will define how our test works. And we look forward to reporting those data. We're confident that we're developing the test with everybody and for everybody from the beginning.
Wonderful. Thanks so much. The next question from the audience is, how is your company's approach to MCED testing different from others?
Well, I think there are several key differences between how we're approaching the development of our MCED test and how others may be approaching. And I think one obvious difference is in the test design. We're committed to a multi-biomarker class approach. We know that cancer secretes a variety of signals into the bloodstream, and we think that complementary information comes from multiple biomarker classes, which means that we're likely to get a more robust and reproducible performance that more closely reflects the biology of the cancers we're trying to detect. So the test design, we believe, is unique. Another unique feature is our approach to diagnostic resolution. Here, we're focused on an imaging-based approach to resolve a positive MCED test. Imaging has been tasked with localizing human disease really since its inception. It's what imaging is all about. And an imaging-based approach in our hands is providing us robust ability to localize cancers where they're present and to rule out the presence of cancer when it's absent. We now have 4-year follow-up from DETECT-A showing us that this approach works very well. It also happens to be an approach that's relatively straightforward to implement. When physicians and healthcare providers are faced with the need to diagnostically evaluate an MCED test result that signals the possibility of cancer, our approach would give them a clear path that they can carry out readily and confidently to get to an answer. So that's another key difference. And I think the third key differentiator for us is that Exact Sciences has a long-standing history of commitment to rock-solid evidence. If you look at the biggest blockbuster products that we offer today, those are Cologuard and Oncotype DX Breast. Those inform the early detection of colorectal cancer and therapy selection for women with breast cancer. And those two tests are backed up by really extraordinarily large and robust studies that have provided really unassailable evidence of their utility. And we're going to do the same thing with MCED as we conduct the largest randomized clinical trial of an MCED in the United States ever done and seek FDA approval for our test. So our commitment to rock-solid evidence that has defined the company really informs how we're going to approach MCED.
Thanks for laying that all out. Sounds like exciting times ahead. Our next question from the audience: what are the watch outs in developing MCED testing, and how is Exact Sciences addressing these?
Well, this is a brand new area, and so I think we're all learning as we go. I mean, the watch outs really start with the test design. It's all about a test that is high sensitivity, high specificity, detects a broad range of cancers, but especially detects solid tumors at early stages. So one of the watch outs is making sure that you've done the work to develop the sort of test that when all is said and done can detect cancers early enough to make a difference. And we're seeing in DETECT-A evidence of that. I shared earlier our long-term follow-up results that show that many of the cancers were detected at early stages, and when they were detected at early stages and treated, the results are exactly what we'd hoped for. So that's a key watch out. Another key watch out is really developing a robust diagnostic strategy that folks can implement. I know we've thought a great deal about it and came to the conclusion that an imaging-based approach is the most efficient way that requires the fewest diagnostic procedures across a population and also happens to be relatively straightforward, so primary care doctors across the country can confidently deploy that. That's how we're addressing the watch out of the possibility of a complicated and meandering diagnostic approach. And I think we've talked about the importance of diversity as a watch out, and that's something that we're deeply committed to. And then, learning how to deploy this test equitably, robustly, and really across the country. We're really focused on that. We think an FDA approval is a linchpin of that. We don't think we can really realistically make an MCED test available broadly, equitably, and across the United States without having an FDA approval and subsequent coverage by multiple healthcare payers. So, a watch out is making sure that your development path has a clear view to how to make this test available to everyone who needs it.
Great. Well, that's all the time we have for today, but thank you so much, Dr. Baer, for sharing your time and insights with us all today. I'd also like to thank Exact Sciences for sponsoring today's webinar. To our audience members, thank you so much for tuning in, and we hope you have a wonderful rest of your day.